So today I'll discuss on DNA replication. DNA replication is a biological process by which a cell makes the exact copies of its DNA. And this process happens in all the living. So this is the DNA double helical structure. Consists of phosphate and sugar backbone. And both these strands are attached with each other via the hydrogen bondings through base pairs. And these bases jutting inside to form the steps of staircase. And while during the course of replication, there is a separation of these two strands, and each strand is capable of generating a newer DNA. So from one DNA strand to two progeny DNA, right? So each strand of the DNA acts as a template for other strand during the replication process. And why we call it is a semi-conservative replication, because once the two progeny strands through two progeny DNAs are being formed, each is having at least one strands available. So that's how this process is known as semi-conservative process. And the pitch of this spiral, uh, the pitch of this spiral is 3.4 nanometer per time. So we from here to here, the distance is 3.4 nanometer. This is the one turn. Then diameter is 1.9 to 2 nanometer. Then protein interact with DNA at exposed bases present on in the two groups. So while no turning over, there are formation of certain groups. These are groups. Few are major groups. If the groove is having more depression, that will be the major group than minor groups. And these are the sites where there will be attachment of proteins, and these proteins control all the regulatory processes of DNA. Right? So these major and minor groups, and these are the parallel to the phosphodiester backbone and wind along the molecule. So these are the sites for synthesis for the attachment of protein, catalytic protein. Now, DNA wraps twice around the histone octamer to form one nucleosome. So this is the pattern how the DNA is packed in the form of a chromosome. So chromosome does consist of nucleosome, and nucleosome is a pattern of attachment or packing of DNA strand with that of histone protein. So we can see this is a histone, these are the histone octamer, and the DNA strand is winding around it. So the core histone as an octamer. Then protein containing the unusually high concentration of basic amino acids. And the 5 classes of histone proteins are there: H1, H2A, H2B, H3, and H4. So these are the various types of histone protein. H1 protein is loosely associated with DNA. Then core histones, the other histones H2A and H2B are lysine-rich histones, and while H3 and H4 are arginine-rich histones, all histones except the H1 are present in equimolar concentration. H1 is in half concentration. And the histone synthesis stops when the DNA synthesis stops. Histone synthesized in the cytoplasm and migrate to the nucleus for packing and for formation of chromatin. Then histones are modified by acetylation, methylation, and phosphorylation. So phosphorylation of serine and threonine residues of H1 occurs prior to the mitosis. Then phosphorylation of H2B is also associated with S phase of the cell cycle. So this is the DNA double helix, which is 2 nanometer thick. And then you can see these are the nucleosome, each of the size of 10 nanometers. So octamer, and around the octamer histones, there is a winding of DNA. Then these nucleosomes attach with each other, packed around each other, they're forming the chromatin fibril, which is around 30 nanometer thick. Then extended chromosome, which is around 300 nanometer thickness. Then this is the chromosome proper. This is how the packing of DNA and histone in the form of chromosome, and its thickness is around 1400 nanometers. So DNA condenses rapidly to form the chromosome. Then what is the melting of DNA? Melting of DNA is known as denaturation, and that leads to the separation of DNA strand by the heat. Optical absorbance of DNA is increased in this process, and that is known as hypochromicity of denaturation. So then melting temperature, the temperature when once there half of the DNA is denatured. Then GC-rich DNA melts at higher temperature because there are higher level of hydrogen bonding in between the G and C base pairs. Then melted strands are reassociated at low temperature. This process is known as annealing. Then formamide is commonly used agent in the DNA hybridization studies, and it lowers the transition melting temperature by disrupting the hydrogen bonding between the bases. Then phosphodiester bonds are not broken in the process. In higher organism, the linear DNA is twisted around its own axis when a supercoil is being formed. And topoisomerase are a kind of enzymes that can relax the supercoil. We will show you in further in the picture how these there is a relaxation of supercoils and how the formation of supercoils takes place. There are two types, one type 1 and type 2 topoisomerases. Gyrases are the enzymes that can insert the supercoils. In higher organism, DNA is organized inside the nucleus. The double-stranded DNA is first bound over the histone. It is called the nucleosome. You have just seen in the previous slide. Then chromatin, it is a long stretch of DNA in association with histones. We have seen the sequential packing of DNA and histone protein in the form of chromatin. Then chromosomes are the condensed chromatin, and extra chromosomal DNA includes the plasmids, which are found in most of the bacteria and other organisms. Then about only 10% of the human DNA contains the gene, and rest are the silent areas. And the segment of gene coding for protein are called as exons, while introns are incapable of, gene expression. They are interspaced in the DNA with the stretches of silent areas. And the primary transcript, we have seen in the transcription process that primary transcript contain the intron sequences, which are later removed to produce the mature messenger RNA. And there is an important role of spliceosome in removal of these introns. Introns are not capable to be translated. And you have seen, I have already told you, this is the process of replication from one DNA to two daughter DNA. So one strand each of, so this is the replication, and you can see that there are two different colored strands, and you can see in both the strand, the one strand in each progeny, and that's how this process is known as semi-conservative process. The replication is always semi-conservative process. A new complementary strand is synthesized over the old template as per the complementarity. Now, new strand is synthesized from 5' to 3' direction, and base pairing rule is always maintained because this is the double helical strand, and always there is a pairing of G with C and A with T, right? So here there is a more clear version, the parent strand, how it is going to be separated out, and the new strand is made and the strand based on the base pairing rule. So if there is a G nucleotide containing the G as a base exposed, the next opposite nucleotide will be having the C as an base. So they are both of these attached via the hydrogen bonding. So on the basis of complementarity, there will be attachment of nucleotides in the newly synthesized DNA. So at the time of replication, how it is happening? So these are the two strand, 5' to 3', 3' to 5'. So in the AT rich region, means the region where there is, abundance of AT sequences available. So in this particular site, there will be certain initiation protein, they will attach themselves, and there will be formation of, adjacent to this side, there will be formation of bubble, means unzipping at the AT rich region of the DNA duplex. So because of unzipping means the removal of hydrogen bonding via certain, with the help of certain protein, detachment of these two strands, leading to the formation of sort of bubbles. And then finally the recruitment of proteins to synthesize the daughter strands. So it is a very complicated process, just a blueprint how in the major steps of formation of bubble, replication bubble, and the role of protein. So let's see further what happens at more complex level. So what are the basic line of differences between the prokaryotic and eukaryotic replication? So origin of replication. Uh, First, the DNA. The DNA in the prokaryote is circular DNA, while in the eukaryote it is linear. So origin of replication is there. Is only one. There is one origin of replication in prokaryotes, while in the eukaryote there are multiple sites of origin of replications. Then single strand binding proteins. SSB, in brief, we call them SSB proteins. So these are the cooperative binding to the ssDNA. Single strand DNA, they bind with each strand, hold them just like fork, so that they get separated with each other. So ssDNA binding proteins bind at the replication fork here in the eukaryotes as well. Then RNA primer, it is the primer which is required for a start of synthesis of DNA. So for DNA, synthesis of new DNA strand, always we require a primer in the form of RNA. So in prokaryote is required for synthesis of both the strands. And while in the eukaryotes, the DNA polymerase alpha has the primase activity and initiates the synthesis of both lagging and leading strands. Then DNA polymer rases. The major polymerizing enzyme in the prokaryote is DNA polymerase type 3, while among the eukaryote, it is the DNA polymerase delta and DNA polymerase epsilon. Keep remember, there are 5 types of DNA polymerase in the eukaryotes, while there are only 3 types of polymerases in the prokaryote. They are type 1, 2, and 3. Then so we will see it here how there is a detachment of two strands with the help of SSB proteins. They are separated and molded by this protein. So a sort of fork is being found. So what is happening here? The two strands are there. One is the leading strand, one is the lagging strand. So in the leading strand, we will see here there is a formation of primase at this particular site, which is away from the replication form. So the synthesis of RNA polymerase, once it is found at this particular site, so it will lead to the continuous synthesis of RNA. Once still here, the synthesis of new strand of DNA happens, then it again opens just like zip opener, and it opens up and further synthesis keeps going on. While in case of lagging strand, we call it a lagging strand because DNA synthesis always takes place from 5 prime to 3 prime direction. Here also it is 5 prime, right from 5 prime to 3 prime direction. Here also it is from 5 prime to 3 prime direction, because in the lagging strand there is a the 5 prime end is here, so definitely the 3 prime end is here. So the new primase will be synthesized somewhere here, and it will lead to the synthesis of from trimase, then further sequential joining of nucleotides with the help of DNA polymerase and formation of DNA strand. Further, it starts from here. Suppose till here the synthesis happened. Now it stops because now we reach to the end. Further, once it is open, then again space is empty. Then new synthesis of primase takes place, and then again there is a stretching of DNA right from 5 prime to 3 prime end. So, and again, it opens up, then synthesis starts new with the help of formation of new primase and primase right from 5 prime to 3 prime end. This is how, due to the direction of the two strands, one is the leading strand, because here opening is right from 3 prime to 5 prime end, and here in the lagging strand opening is from 3 prime to 5 prime end. So this directionality leads to the formation of continuous and discontinuous synthesis of DNA. And here in the lagging strand, we can see there are small fragments of DNA formed. So these are strands are known as discontinuous strands, or these are known as Okazaki fragments, based on the name of explorer who for the first time saw the formation of these small strands of DNA in the lagging strand, right? So we can see it here. And once there is a unzipping, there is a continuous opening, slowly opening with the formation of numerous strands of DNA. So once it opens and the two strands keep holded by the SSB proteins, so there will be a supercoiling in the outer side, in the double helical side. So this leads to the strain, and these supercoils they are going to be relaxed or relieved with the help of enzyme topoisomerases. So these topoisomerases type 1 and 2, what do they do? They cut the strands, relieve the supercoils, and then rejoin and ligate them. So this is how there is a facilitation of opening of these double strand into the single strand. Understood? So then we will see here the more clear account. So primase synthesizes an RNA primer over the parent DNA. So first, for the formation of before the formation of before the action of DNA polymerase, there should be a synthesis of primer. So primer synthesis takes place with the help of enzyme primase. So these are few base sequences of nucleotides, and a small fragment of RNA is being formed. And over this fragment, then there will be attachment of DNA polymerase. Primase is removed, and at this place, there is attachment of DNA polymerase. And then from this, from 5 prime end to 3 prime end, there will this green color signal shows that there is a now DNA synthesis is being taking place with the help of DNA polymerase. And this, as based on the complementarity of the template strand, there will be formation and joining of nucleotides, leading to the formation of newer strand of DNA, right from 5 prime to 3 prime direction. So in the next step, you see here, a new DNA polymerase removes the RNA and replaces with DNA. So you have seen it here, a new DNA polymerase removes the RNA. This polymerase, finally, it will remove the RNA, which was being formed this time, and it is now replaced with the newer DNA synthesis. Primase role is only to act as a platform on which the DNA polymerase will attach, and this will further lengthen the newer strand of DNA. Further, once its function is over, this RNA has to be removed with the help of another type of DNA polymerase. So Then ligase joins the end, and now the daughter strand is now complete. So you have seen in the lagging strand, a lot of nicks are there. These nicks are to be sealed with the help of enzyme ligases. So there are certain DNA polymerases in the prokaryotes and the eukaryotes. They perform the function of ligation. So this lead to continuous formation of DNA strand right from the discontinuous fragments. That's all about the transcription.